Welcome to our microbiome analysis training module. In this cloud-based module, we'll take you through the process of analyzing microbiome sequencing data, from raw data to meaningful insights. Whether you're new to microbiome bioinformatics or looking to refine your workflow, this training will provide you with the essential skills needed to work with microbiome data. This training module is divided into 3 submodules. The first submodule provides a comprehensive background on 16S rRNA sequencing, covering its principles and applications in microbiome research. In the second submodule, we'll process raw sequencing data, ensuring quality control and preparing it for analysis. Finally, the third submodule integrates survey-based metadata, allowing us to explore associations between the gut microbiome, diet, and lifestyle factors. Before we dive into the analysis, let's first discuss why microbiome research is important and how we collect and process microbiome data, and what you will learn in this training. Microbiomes are microbial communities that play essential roles in human health, environmental ecosystems, and disease. Microbiomes exist in many forms, but we'll focus on the gut microbiome in this module. However, the techniques you will be taught apply to all microbiome studies. The gut microbiome influences our digestion, immune function, and even mental health. Understanding microbiome composition can help us answer key questions such as, how does diet shape the gut microbiome? How do external factors such as sex and body mass index influence microbiome composition? To answer these questions, we'll use 16S rRNA sequencing. This approach leverages the 16S rRNA gene, a highly conserved genetic marker found in all bacteria. The 16S ribosomal RNA, or rRNA, is a component of the small subunit of the prokaryotic ribosome, playing a critical role in protein synthesis. Its gene is comprised of both conserved and hypervariable regions. The conserved regions allow for the design of universal primers to amplify the gene across diverse bacterial species, while the hypervariable regions provide unique sequences that enable taxonomic classification. By sequencing these variable regions, we can distinguish and classify bacteria, often down to the genus or species level, providing critical insights into the community composition and diversity of our samples. We can consider the 16S gene as a bacterial fingerprint. The first step in microbiome analysis is sample collection. Depending on the study, samples can come from a variety of sources: stool samples for gut microbiome research, skin swabs, soil, or even water. The goal is to capture the bacterial communities present in a particular environment for analysis. Once we have the samples, the next step is DNA extraction, which includes purification steps. This process isolates all genetic material from the microbes in the sample. After extracting the DNA, we use PCR amplification to target a specific gene, in this case, the 16S rRNA gene. Once the target DNA is amplified, we prepare it for sequencing by adding adapters and barcodes. This library preparation step allows us to sequence multiple samples in a single run while keeping track of which sequences belong to which samples. With the samples prepped, we moved on to DNA sequencing using a platform like Illumina. This process generates FASTQ files which contain raw sequence reads along with quality scores. At this stage, the data is still messy. It includes sequencing errors, merged sequences, and other artifacts that need to be removed before we can make meaningful conclusions. This is where bioinformatics comes in, and where our module will start. In submodule 2, we will focus on processing raw FASTQ files using the Data2 package to prepare our sequencing data for analysis. This includes quality filtering, error correction, and removal of merged sequences to ensure that only high-confidence sequences are retained. We will then identify unique amplicon sequence variants, ASVs, and create a table that represents the microbial composition of our samples. By the end of this submodule, we will have a clean, processed data set that is ready for taxonomic classification and downstream microbiome analysis. In submodule 3, we will integrate metadata and perform in-depth microbiome analysis. Microbiome data becomes far more meaningful when combined with metadata, information about the samples, such as dietary habits, lifestyle factors, and physiological characteristics. This helps us understand how different factors may shape the gut microbiome. We will start by importing metadata collected through participant surveys. These surveys contain information such as dietary intake, physical activity, and demographic details like age, sex, and weight. Once we have reviewed the metadata, we will use a package called PhyloSeq to analyze our microbial ecology. We will classify the microbiome and explore diversity, determining how different dietary and lifestyle factors play a role. If you are new to microbiome research or bioinformatics, don't worry. This training module provides step-by-step guidance to help you navigate the process. Microbiome science is a rapidly growing field, and learning how to analyze sequencing data is an invaluable skill for researchers across disciplines. By the end of this module, you'll have an understanding of microbiome research, how to analyze and explore these data sets, and how to interpret diversity plots. Let's get started.